Hi, everybody. Josiah Gilliam here, Executive Director at Pump, and we're here with another community partner highlight about our exciting collaboration with the Hillman Cancer Center on Science, Chilled, and Distilled, an event series that we've been working on, where we bring team members from the Hillman Cancer Centers, researchers, caregivers, to talk about the life-saving and life-changing work that they're doing in our region, in unique locations around the area, and we invite community into that. And we're thrilled to have another coming up on the 20th of this month at Trace Brewery. And we're thrilled to have two team members from the Hillman Cancer Center with us today. We're going to have a little bit of a conversation about the work that they do, about this series, about what folks can expect at Trace later this month. Thanks so much for being here, guys.
Thank you for having us.
Yes. Thrilled. Let's start with introductions, and then we'll get a little bit more into the details. So please, who you are and what brought you to the Hillman Cancer Center.
Okay. I'm Patty Opresko. I'm a professor of pharmacology and chemical biology at the University of Pittsburgh School of Medicine, and I'm co-leader of the Genome Stability Program at the Hillman Cancer Center. So my lab just turned 20. I'm very proud of that.
Congrats.
It's a big milestone. Yeah, thank you. So I joined the Hillman Cancer Center, University of Pittsburgh, 20 years ago. And what really brought me here was a growing group of scientists that study DNA damage and repair, something I've been very passionate about for years. So I saw this as an opportunity to work with other scientists, to learn from other scientists. And I had heard from colleagues in the field that Pittsburgh was a very collaborative community, and I have found that to be true. And for those reasons, my science has really thrived over the years. And I'm grateful for the opportunity to interact with other scientists at the Hillman and in Pittsburgh in general.
That's wonderful. Thanks for being here.
My name is Dayana Rivadeneira, and I am a brand new assistant professor at Hillman Cancer Center and part of the immunology department. And yeah, I just started my lab 4 months ago. It's been a really exciting opportunity, and I think is a testament to the opportunity that Pittsburgh gives you of growing as a young scientist. Hillman Cancer Center has been a great place to be. I've been there for about 9 years with my husband, where we moved from Philly, and we were very pleasantly surprised to what we found, and we've been here ever since. And it's been, like Patty mentioned, a great environment for collaboration and just being able to launch your career as a young scientist. And this is how I met Patty. So we started a collaboration a few years ago on a project that has been really taken off and has been my launching pad for my career as well. So it's a testament to the environment that we have at the Hillman. So it's been really nice.
Congrats on your lab. That's a big
Thank you.
deal. That's
Thank
wonderful. So we don't want to spoil the presentations on the 20th, but let's do a little bit of learning now, kind of in the moment. So give us some high-level definitions just for people that might not be familiar, myself included, to be fair. When you say immunology, how do you describe, what is that term? How should people think about that?
I mean, you should think about, the first line of safety in your body for anything, really, external, internal, any kind of exposure of any kind of pathogen. You can envision your body mounts an immune response, and it can really take care of dust mites to cancer. So you really have to think about how you can, harness that immune system and how it can help you just combat any kind of disease. We have, a whole history of COVID. Your immune system was very important. In the Hillman Cancer Center, we think about how to harness that immune system for treatments of different types of cancers. So that's, in general, how you can think about it.
I love it. From dust mites to cancer.
Dust mites from cancer.
That's so terrific. Genome stability. Help us understand that.
Yeah. So what's also very interesting about the immune system is broken pieces of DNA and damaged DNA can also activate the immune system. So this is something that's led me to my interactions with Dayana and other individuals in the immunology department. So genome stability, what does that mean? Well, our genome is the blueprint for life. So it's incredibly important for regenerating cells, healthy cells that are exact copies of our functioning cells within our body. It's very important for producing the types of proteins and things that our cells need to function. If you think of a blueprint, what things do you need for a building to function? All of that information is in our genome. But our genome is also susceptible to damage. So one of the clearest examples is UV light. You go out into the sunlight, that sunlight can react with the DNA. The base is in our genome, ATCG. You might remember these from biology class, right? And can change the chemistry, the makeup of these bases, and essentially damage them. And that then can lead to changes in the genome that can eventually convert that normal healthy cell into a cell that is now cancerous and can grow into a tumor. And so this is why it's so important to wear your sunscreen. Other factors can also damage our genomes, such as chemotherapeutic agents, things that we use to treat tumors and cancers. And that's because if those tumor cells, if their genomes are damaged, they can't function properly, then they can't divide and they'll die. So the goal is to really target and kill the bad guys, the tumor cells, right? In the meantime, how can we preserve our healthy cells? Things like our immune cells, right? So it's very important to understand how our genome is damaged, what are the consequences of that damage, and how that damage is repaired.
Another quick question about this, because as you guys are sharing, about these terms, my mind goes to kind of, like global and then minute, I guess. So there are things maybe we can do to support our health systemically, the systems in our body systemically. But then I hear you speaking about fragments of DNA, about a tumor, for example. How do you both think about that, I guess? And how specific are we able to get at this point with the science on a specific, say, tumor or event that's happening within the body? If that makes sense.
Okay.
I guess the way that I could think about it, I would say yes on both things.
Okay.
We can think about it in a very broad sense of now. In the Cancer Center, we've been thinking a lot about all the different ways that the DNA can be affected, our immune cells, like how you eat, when you sleep, how many hours you get of sleep, what you're eating. We heard from our last event, the microbiome is important. What you eat is who you are. And so you can think about it in big picture. But then we have also, science has advanced so much that we have the capacity to really go down to the very smallest nitty-gritty like aspects of the genome and how the mechanisms are being formed that you can harness and change for you to analyze how is the genome being broken into tiny little pieces and how we can prevent it from breaking down. So really we can go in and analyze it to the as specific as we can, but we can also do things in the more approachable way as to what you're eating and are you exercising or you're not exercising. So we can do it both ways, and we do it both at the Hillman.
Yeah. So we, in the genome stability field too, we think a lot about diet and exercise, our environment, how do things that we're exposed to, like sunlight, like pollution, affect our genome? What are things that we can do to protect the genome in our healthy cells so they don't acquire damage that can lead to genomic changes and mutations that can cause cancer? So things like eating foods rich in antioxidants. So we know free radicals, oxidative stress can damage, just like sunlight can damage our genome, things like chronic inflammation. So it's in the genome stability field, we think about how can we protect our genome by eating healthy. We know exercise can also help protect our genome. So how do we look at a granular level? At the Cancer Center, we think about developing biomarkers for DNA damage. So how can we look at to see whether or not damage has been accumulating? Some of that is occurring now through high-level next generation sequencing. But we also think too about those tumor and cancer cells. What types of drugs can we use to damage their genome? Because we know that these tumor and cancer cells acquire mutations and changes, and some of those mutations affect the way these cancer cells can repair their genome. So they might acquire a mutation in a gene that encodes for a protein that allows them to fix and repair their genome. So some people refer to that as an Achilles heel. The bottom line is, how do we protect the good guys, right? And how do we take advantage or leverage some changes in the bad guys, the cancer cells and the tumors, to make them more sensitive to DNA damaging agents.
Wonderful. Well, my mom's gonna love this part of the of the interview. 'Cause she's always encouraging sunscreen application, and I need to reapply as well. if that's right.
Reapplication, please.
Reapplication, please. Yeah,
Every 2 hours.
that's terrific. And
If
it's
you're
good
sweating, more reapplication.
every 2 hours, and if you're sweating more.
Exactly, because you're sweating your sunblock away.
I see.
Unless it's waterproof. Anyway, I
Gotcha.
digress. I feel very strongly about sunblock.
That's wonderful. Yeah. Well, it's a best practice, right? And a way to shield the body and to support the good guys. It sounds like, and mitigate the chances of the bad guys, so to speak, and all of that. And we're thrilled to hear that too, because at Pump, wellness is important to us, and through the PSL, our largest program, Pittsburgh Sports Leagues, we're providing so many opportunities for people to exercise and experience that good quality of life in that way. And we don't necessarily tie it to cancer prevention or something like that, but it is a good thing to have in your life. So that's really great. Let's give people a primer on what they can expect at Trace. What will the presentations kind of focus on? I know we've touched on it a little bit, but what can people expect when they come see us on January 20th?
So I can start out. My lab has really been focused on a very special part of the genome for the past 20 years. So we're very interested in the little caps, the tips at the ends of our chromosomes called telomeres. This is because telomeres are so important for both cancer and aging, and you'll hear about some of this in our presentation at Trace. Studies over many, many years have found that there's a strong correlation between oxidative stress and accelerated shortening of these telomeres, or uncapping of these telomeres to the point where they can't really function anymore. And so we really wanted to understand why, right? Why is that? What is it about the telomeres that make them so sensitive to oxidative stress? And how could we move beyond correlation to get at cause? Is it because the telomeres are being damaged? And if so, does the cell care, right? What consequence does that have for the ability of those telomeres to cap the ends of the chromosomes? They can't do it as well. How does that affect normal cells? How does that affect cancer cells and
Immune cells.
Immune cells.
And that's kind of where I come in. So linking a little bit to what Patty was saying, oxidative damage is very important also in tumors, and it's seen a lot by different types of solid tumors. And as we know, the immune system is important for the battling cancers, and we found that immune cells can also experience that oxidative damage, can affect their telomere function, and that's what we're going to be going into a little bit in our presentation, and how that telomere changes based on that exposure in the tumor can affect their function against cancer, and how we can prevent it. Is there a way that we can prevent that damage and improve an immune response against cancers? That's really the goal, and that's where I kind of get in there in the process. So Patty is the expert on all things telomeres, and so she's been.
And I know little about the immune system, but I'm learning.
So we kind of have like a great team in that sense, where I bring in an immune expertise and she brings a telomere expertise, and we've been working together.
Yeah, and I think that is something that really makes the Hillman Cancer Center very special. We're an NCI designated comprehensive cancer center. And so what that means is we receive funding from the National Cancer Institute to really support infrastructure, to support mechanisms, to bring scientists at different stages of their careers, in different disciplines, in different facets of, we have scientists that are working the basic science level, trying to address fundamental questions. We have scientists working more at the translational level. How do you translate those discoveries at the bench into therapeutics? And then we have clinicians, right, that we can partner with. So I think being able to partner with scientists at different disciplines and at different levels is really where the magic happens. I never thought I would be studying T cells or immune cells, but it turns out...
Or me telomeres.
Right. But it turns out there's a lot of crosstalk and
absolutely.
a lot of interaction. So
Wonderful. Well, it sounds like a great team, and a perfect example of that collaboration at Hillman and in our region that's being fostered. Okay, so Trace Brewery, the 20th of January, 5:30 start time. There will be refreshments for folks, and a terrific venue. We're really excited to be working with Trace, which is a wonderful community venue in so many ways. So we hope to see you all there. Thanks so much for checking out this video, and we'll see you next time.